Leaked grant proposal details high-risk corona research. The proposal, rejected by U.S. military research agency DARPA, describes the insertion of human-specific cleavage sites into SARS-related bat coronaviruses. A grant proposal written by the U.S.-based nonprofit EcoHealth Alliance and submitted in 2018 to the Defense Advanced Research Projects Agency, or DARPA, provides evidence that the group was working, or at least planning to work, on several risky areas of research. Among the scientific tasks the group described in its proposal, which was rejected by DARPA, was the creation of full-length infectious clones of bat SARS-related coronaviruses and the insertion of a tiny part of the virus known as a proteolytic cleavage site into bat coronaviruses. Of particular interest was a type of cleavage site able to interact with furin, an enzyme expressed in human cells. The EcoHealth Alliance did not respond to inquiries about the document, despite having answered previous queries from The Intercept about the group's government-funded coronavirus research. The group's president, Peter Daszak, acknowledged the public discussion of an unfunded EcoHealth proposal in a tweet on Saturday. He did not dispute its authenticity. Since the genetic code of the coronavirus that caused the pandemic was first sequenced, scientists have puzzled over the furin cleavage site. This strange feature on the spike protein of the virus has never been seen in SARS-related beta coronaviruses, the class to which SARS-CoV-2, the coronavirus that causes the respiratory illness COVID-19, belongs. The furin cleavage site enables the virus to more efficiently bind to and release its genetic material into a human cell, and is one of the reasons that the virus is so easily transmissible and harmful. But scientists are divided over how this particular site wound up in the virus, and the cleavage site became a major focus of the heated debate over the origins of the pandemic. Many who believe that the virus that caused the pandemic emerged from a lab have pointed out that it is unlikely that the particular sequence of amino acids that make up the furin cleavage site would have occurred naturally. Adherents of the idea that SARS-CoV-2 emerged from a natural spillover from animal hosts have argued that it could have evolved naturally from an as yet undiscovered virus. Further, they argued scientists were unlikely to have engineered the feature. There is no logical reason why an engineered virus would utilize such a suboptimal furin cleavage site, which would entail such an unusual and needlessly complex feat of genetic engineering. 23 scientists wrote earlier this month in an article in the journal Cell, there is no evidence of prior research at the Wuhan Institute of Virology involving the artificial insertion of complete furin cleavage sites into coronaviruses, but the proposal describes the process of looking for novel furin cleavage sites in bat coronaviruses. The scientists had sampled and inserting them into the spikes of SARS-related viruses in the laboratory. We will introduce appropriate human-specific cleavage sites and evaluate growth potential in a type of mammalian cell commonly used in microbiology and HAE cultures, referring to cells found in the lining of human airways, the proposal states. The new proposal, which also describes a plan to mass vaccinate bats in caves, does not provide conclusive evidence that the virus that caused the pandemic emerged from a lab, and virus experts remain sharply divided over its origins, yet several scientists who worked with the coronavirus told The Intercept that they felt that the proposal shifted the terrain of the debate. Then here's the entire diffused proposal, tipping the scales. Some kind of threshold has been crossed, said Alina Chan, a Boston-based scientist and co-author of the upcoming book Viral: The Search for the Origin of COVID-19. Chan has been vocal about the need to thoroughly investigate the possibility that SARS-CoV-2 emerged from a lab, while remaining open to both possible theories of its development. For Chan, the revelation from the proposal was the description of the insertion of the novel furin cleavage site into bat coronaviruses, something people previously speculated but had no evidence may have happened. Let's look at the big picture. A novel SARS coronavirus emerges in Wuhan with a novel cleavage site in it. We now have a pandemic. In the early 2018, they had pitched inserting a novel cleavage site into novel SARS related viruses in their lab, said Chan. This definitely tips the scales for me, and I think it should do that for many other scientists as well. Richard Ebright, a molecular biologist at Rutgers University who has espoused the possibility that SARS CoV 2 may have originated in a lab, agreed. The relevance of this is that SARS-CoV-2, the pandemic virus, is the only virus in its entire genus of SARS-related coronaviruses that contains a fully functional cleavage site at the S1 and S2 junction, said Ebright, referring to the place where the two subunits of the spike protein meet. And here's a proposal from the beginning of 2018 proposing explicitly to engineer that sequence at the position in chimeric lab-generated coronaviruses. Martin Wikelski, a director of the Max Planck Institute of Animal Behavior in Germany, whose work tracking bats and other animals was referenced in the grant application without his knowledge, also said it made him more open to the idea the pandemic may have roots in a lab. The information in the proposal certainly changes my thoughts about a possible origin of SARS-CoV-2, Wikelski told The Intercept. In fact, the possible transmission chain is now logically consistent, which it was not before I read the proposal. But others insisted that the research posed little or no threat and pointed out that the proposal called for most of the genetic engineering work to be done in North Carolina rather than China. Given that the work wasn't funded and wasn't proposed to take place in Wuhan anyway, it's hard to assess any bearing on the origin of SARS-CoV-2. Stephen Goldstein, a scientist who studies the evolution of viral genes at the University of Utah and an author of the recent Cell article, wrote in an email to The Intercept. Other scientists contacted by The Intercept noted that there is published evidence that the Wuhan Institute of Virology was already engaged in some of the genetic engineering work described in the proposal and that viruses designed in North Carolina could easily be used in China. The mail is filled with little envelopes with plasmid dried to the filter paper that scientists routinely send each other, said Jack Nunberg, director of the Montana Biotechnology Center at the University of Montana. Vincent Ricanelio, a professor at microbiology and immunology at Columbia University, was adamant that the proposal did not change his opinion that the pandemic was caused by natural spillover from animals to humans. There is zero data to support a lab origin notion, Ratcanello wrote in an email. He said he believed that the research being proposed had the potential to fall into the category of gain-of-function research of concern, as did an experiment that was detailed in another grant proposal recently obtained by The Intercept. The government funds such research in which scientists intentionally make viruses more pathogenic or transmissible in order to study them only in a narrow range of circumstances. DARPA rejected the proposal, at least in part because of concerns that it involved such research. While Ratcanello acknowledged that the research in the DARPA proposal entailed some danger, he said that the benefits far, far outweigh the risk. He also said the fact that the viruses described in the proposal were not known pathogens mitigated the concern. This is not SARS, he said, referring to SARS-CoV-1, the virus that caused the 2003 outbreak. It's SARS-related. But SARS-CoV-2 is not a direct descendant of that virus. It's a relative. In fact, the viruses described in the grant proposal, which was first posted online by the research group Drastic, were not known pathogens, and the authors of the grant proposal make the case that because the scientists would be using SARS-related bat virus as opposed to the SARS virus that was known to affect humans, the research was exempt from gain-of-function concerns. But according to several scientists interviewed by The Intercept, the viruses presented a threat nevertheless. The work describes generating full-length bat SARS-related coronaviruses that are thought to pose a risk of human spillover. And that's the type of work that people could plausibly postulate could have led to a lab-associated origin of SARS-CoV-2, said Jesse Bloom, a professor at Fred Hutchinson's Cancer Research and director of the Bloom Lab, which studies the evolution of viruses. Bloom pointed out that the scientists acknowledged the risk to humans in their proposal. It's an explicit goal of the grant to identify the bat SARS-related coronaviruses that they think pose the highest risk. Stuart Norman, a professor of cell biology who directs the developmental biology laboratory at New York Medical College, also said the fact that the viruses weren't known to be dangerous didn't preclude the possibility that they might become so. That's really disingenuous, Newman said of the argument. The people that are claiming natural emergence said that it begins with a bat virus that evolved to be compatible with humans. If you use that logic, then this virus could be a threat because it could also make that transmission. Newman, a longtime critic of gain-of-function research and founder of the Council for Responsible Genetics, said that the proposal confirmed some of his worst fears. This is not like slightly stepping over the line, said Newman. This is doing everything that people say is going to cause the pandemic if you do it. While the grant proposal does not provide a smoking gun for the SARS-CoV-2 escape from the lab, for some scientists it adds to the evidence that it might have. Said Nunberg of Montana's Biotechnology Center. Once you make an unnatural virus, you're basically setting it up in an unstable evolutionary place. The virus is going to undergo a whole bunch of changes to try and cope with its imperfections. So, who knows what will come of it? The risks of such research are profound and irreversible, he said. You can't call back the virus once you release it into the environment. DARPA, a division of the Department of Defense, said regulations prevented it from confirming that it had reviewed the proposal. Since EcoHealth Alliance may or may not be the direct source of the material in question, and we are precluded by federal acquisition regulations from divulging bidders or any associated proposal details, we recommend that you reach out to them to confirm the document's authenticity, a DARPA spokesman wrote in an email to The Intercept. The British Daily Telegraph reported that it had confirmed the document's legitimacy with a former member of the Trump administration. The Telegraph story erroneously reported that the scientists proposed to inoculate bats with live viruses. In fact, they hoped to inoculate them with chimeric S protein, which was proposed to be developed through a subcontract in the Grant and Ralph Barick's lab at the University of North Carolina at Chapel Hill, not in Wuhan. Barick did not respond to the Intercept's request for comment. Conflict of interest. Many questions remain about the proposal, including whether any of the research described in it was completed. Even without DARPA funding, there were many other potential ways to pay for the experiments. As scientists interviewed for the article agreed that often researchers do some of the science they describe in proposals before or after they submit them. This was a highly funded group of researchers that wouldn't let one rejection halt their work, said Chan, the viral author. Perhaps the most troubling question about the proposal is why, within the small group of scientists who have been searching for information that could shed light on the origins of the pandemic, there has been apparently so little awareness of the planned work until now. Peter Daszak and Linfa Wang, two of the researchers who submitted the proposal, did not previously acknowledge it. Daszak, the EcoHealth Alliance president, has actively sought to quash interest in the idea that the novel coronavirus originated in a lab. In February 2020, as the pandemic began to grip major cities in the U.S., he began organizing scientists to write an open letter that was published in The Lancet addressing the origins of the virus. The rapid, open, and transparent sharing of data on this outbreak is now being threatened by rumors and misinformation around its origin, read the statement signed by Daszak and 26 co-authors. We stand together to strongly condemn conspiracy theories suggesting that COVID-19 does not have a natural origin. Daszak directed and gathered signatures for the letter, all while suggesting that he and his collaborators on the proposed DARPA project, Barick and Wang, distanced themselves from the effort. I spoke with Linfa Wang last night about the statement we sent round. He thinks, and I agree with him, that you, me, and him should not sign the statement so it has distance from us and therefore doesn't work in a counterproductive way, Daszak wrote to Barick in February of 2020, just weeks before it appeared in the journal, according to an email that surfaced a year later by Public Health Investigation. investigative research group U.S. Right to Know. We'll then put it out in a way that doesn't link it back to our collaboration so we maximize an independent voice. Ultimately, Daszak did sign a letter. I also think this is a good decision, Barrett replied. Otherwise, it looks self-serving and we lose impact. Barrett M. Wang, a professor in emerging infectious disease program at the Duke NUS Medical School of Singapore, did not respond to inquiries from The Intercept about their decision not to sign the letter in The Lancet. Daszak was also a member of the joint team of the World Health Organization sent to China in February 2020 to investigate the origins of the pandemic, which concluded that it was extremely unlikely that a virus had been released from the lab. In March, the World Health Organization called for further investigation of the origins of the virus and stated that all hypotheses remain open. I find it really disappointing that one of the members of the joint WHO China team, which is essentially the group of scientists that were tasked as representatives of both the scientific community and the World Health Organization of investigating this, are actually on this proposal, knew that this line of research was at least under consideration and didn't mention it at all, said Bloom of Fred Hutch. Whatever information relates to help people think about this needs to be available, transparently available, and explained. Correction, September 21st, 2021. A previous version of the article stated incorrectly that EcoHealth Alliance proposal had been featured on Sky News Australia. Correction, September 23rd, 2021, 3:30. A previous version of the article stated incorrectly that Linfa Wang was a member of the WHO China team.